Good day, dear students. Our task for today is to talk and to discuss the basis of the life on our planet. We need to talk about structure, organization, and evolution of genetic material in microorganisms. And first of all, of course, I really hope that you all saved your knowledges on genetics from first course biology, maybe from school course. You all know and remember principle of organization of our genome, and of course genome of bacteria and viruses organized in the same manner, the same types of nucleic acids are used. It is deoxyribonucleic acid or DNA and ribonucleic acid or RNA, and they differ by few properties. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. DNA is uses nucleobases, four nucleobases: adenine, thymine, cytosine, and guanine. And RNA uses uracil instead of thymine. So uracil differs from thymine in that it lacks a methyl group on its ring. The main structure of molecules are very similar, and they of course have the same function. So all nucleobases are complementary and usually abbreviated till first letter. So ATCG and U is usually used in literature and papers to identify those nucleobases. And again, common property in eukaryotic cell and the same in bacteria. DNA is presented by double-stranded molecule, RNA is presented by single-stranded molecule. And according to functions, RNA is subdivided into messenger RNA, transport RNA, and etc. Again, I hope you all know it. And DNA is a storage of genetic material. It is used to keep genome. It is more stable molecule, and DNA is transferred to further generations. So, uh, if you talk about organization of genetic material in bacteria and viruses, uh, both microorganisms differs from eukaryotes and differs from one each other. So viruses are very unique organisms. They are pre-cellular organisms, and for virus, it's not obligate to have DNA as main material. And we said already in previous lectures when we discussed viruses that viruses have only one type of nucleic acid. It can be DNA or RNA, and both DNA and RNA can be single-stranded or can be double-stranded. And if virus have single-stranded RNA, it can be negative or positive sense. Positive sense RNA virus means that genome of virus is identical to messenger RNA. If RNA of the virus can be used as a messenger RNA, it is positive sense virus. If RNA of the virus is complementary to messenger RNA, that virus is negative sense. And viruses themselves, they are non-active particles. They can only infect a host cell, and host cell will use genome of virus instead of its own, thinking that it is genome of the cell. By viruses, they cannot use their genome. They only carries it, and actually that's all. The production occurs by use of a host cell. Bacteria is more close to eukaryotes by structure of genetic material. Genome is always presented by double-stranded DNA. That double-stranded DNA is organized in a chromosome, and chromosome is kept as the storage of genetic material. In bacteria, in the contrast to eukaryotes, chromosome is always one, it is haploid, and it's not linear, it is circular. But the main function is absolutely the same. And bacteria can have additional type of genetic information presented as a plasmid. We will talk later in current lecture about structure and functions of plasmids, but for now you need to know that bacteria can have two types of storage of genetic material. It is chromosome and plasmid. Both are double-stranded DNA. So, of course, you all know, you all understand, all know about evolution, and you know that bacteria, as any other alive organism, need to needs to be able to change its property according to or together with changing environmental conditions. Any alive form, any alive organism on our planet should undergoes evolution because environment is changing, conditions surrounding us are changing, and of course, bacteria, any alive organism, need to adapt to new changing conditions. And you all remember that bacteria can multiply only by asexual reproduction. It can multiply only by binary fission. And if you remember that scheme, that process, every new daughter cell will receive identical copy of maternal DNA. And some attentive students can ask how it is possible for bacteria to change genome if bacteria during multiplication cannot change genome. It is always stable, molecule is identical. And of course, for bacteria, it's impossible to have sexual reproduction. You cannot exchange genome with other bacteria during reproduction. But anyway, bacteria have even three processes, three options to exchange its genome. And now we will talk about those mechanisms. So the most common mechanism, the most frequent mechanism, it is a conjugation. Conjugation, it is the process when one bacterium transfers genetic material to another by direct contact. We cannot call it sexual contact, but it is direct contact. One bacterium, and actually in some papers you can find name male bacterium and female bacterium. I don't really like that type of identification because bacteria have no gender, and you all know that bacteria is not a sexual form of life. They all are identical by structure, and it's more correct to say donor bacteria and recipient bacteria. So during conjugation, one bacterium will act as the donor of genetic material, another will act as the recipient. So the main difference between donor and recipient bacteria is that donor carries a DNA sequence which is called a fertility factor or F factor. That F factor allows donor to produce an F pili. As you can see on that picture, one bacterium looking like covered with hairs, one is naked. So that pileated bacteria, besides common pili, they are seen as a black, have one very long, thin, tube-like pili of some orange or pink color. It is F pili. That F pili is used by donor to contact the recipient. Actually, it will connect two cells, and F pili is a tube-like, means it have empty cavity inside. It is a channel, a bridge between two bacteria. And when two bacteria are connected with the help of F pili, F pili draws both bacteria together, and donor bacterium transfers genetic material to recipient bacteria. And usually, bacteria can exchange plasmids. Typically, it is a plasmid which is introduced into the recipient bacteria. And after conjugation, both microorganisms can obtain new properties. So it is the first type of exchange of genetic material, physical direct contact between two bacteria with the help of F pili, which is known as a conjugation. In natural condition, it is the most common mechanism of bacterial evolution. So second mechanism is known as transduction. Partially, you're already familiar with that process. We described it when we discussed bacteriophages. So transduction, it is the process when bacteriophage transfers genetic material from one bacterium to another bacterium. So we know that bacteriophage, it is a virus of bacteria. It can insert its own. We have lytic and lysogenic bacteriophages. Lysogenic bacteriophage can insert genome of itself of virus into bacterial chromosome. It remains latent for some time period, and after that, when it will be activated, it should cut off genome of itself from bacteria, and bacterial and viral genome became separated. But we said that process of that exertion is not very good controlled by enzymes and have poor system of control. And very often, phage can occasionally remove a portion of bacterial DNA and take it within. And bacteriophage will have its own gene plus one of your genes of bacteria. Of course, bacteriophage will not use the gene. Bacteriophage cannot use the genome. But when new bacteria will be infected and genome of the phage will be integrated into the chromosome, bacteria can acquire new genes of previous bacteria, and bacteria will have new property. And we have two types of transduction: generalized and specialized. Generalized transduction, it is a phenomenon when bacteriophage can pick up any portion of DNA and insert it to any accidental locus of bacterial chromosome. And that type of transduction is common in life and nature, but it's not very interesting for us because that type of transduction cannot be predicted. We cannot know which gene will be cut off and where that gene will be inserted. But second type of transduction, specialized transduction, it is a phenomenon when only specific portion of DNA will be carried by virus and it will be inserted into known locus of bacterial chromosome. And that phenomenon of specialized transduction is highly used in science, which is known as genetic engineering. Genetic engineering uses bacteriophages to stably introduce genes of interest into various host cells with the help of viruses or the help of bacteriophages. Gene of interest, I mean gene which is interested for us. Of course, not for bacteria, not for phages. It is something for us. And of course, we use everything only for our purposes, and we can use bacteria and virus to reach some our goals. And if you will read any medical book for medical students, it is almost always the classic example of use of transduction in genetic engineering. It is an E. coli secreting human insulin. You all know disease diabetes mellitus. It is the state when subject cannot produce sufficient amount of insulin, or it cannot produce it in correct time period. And insulin is a hormone required to control blood glucose level. And when people eating, level of glucose is elevated, it needs to be controlled by insulin. If insulin cannot be produced by human, level of glucose cannot be controlled. It can lead to serious consequences, to serious complications, and even lethal outcomes in people who are suffering with severe forms of diabetes mellitus. So we have multiple different drugs, but they not all can help in severe cases, and usually severe cases of diabetes mellitus should be controlled by external insulin provided in injections. So the very first question and the very first problem, where to take that external insulin? Previous decades, that problem was solved by use of animals. Pharmaceutical companies bought organs of animals in some butcher farms and fabrics, and those organs were specifically treated. Insulin was extracted. It was kind of purified, and used for external injections for humans, for diabetic patients. But method is kind of is looking like simple, available, but we had three major problems of that animal insulin. First of all, it is still foreign substance. Molecule of insulin itself is very complex and it is large in size, and it's difficult to purify it very good. And a lot of people who injected animals insulin, they had allergic reactions because it is foreign substance. It's not perfectly purified, and a lot of animal proteins and other animal tissues got into that insulin, and people had allergy. Of course, when it is the only chance for people, it's difficult to use those drugs, and alternatives are not always available and not always good. Second problem, it is a price because company need to buy organs of animals. They are still kind of expensive when we'll talk about organs in huge mass. Of course, it is difficult to store. They have very short storage duration period. It's difficult to deliver them. It is expensive. We need specific, again, equipment to perform that extraction. And still, that method is not very, it's not as simple as it's looking like. The third problem, and nowadays it is again very important problem, it is ethic problem, because actually in Russia it's not very popular, but in foreign countries, especially for example in USA, a lot of people protesting, in Europe as well, a lot of people protesting against killing animals because animal is alive, clever, pain. It is similar to humans, and they uh, have some, they should have some rights. And of course people trying to save lives even of animals in our planet. And the main argument of those people was, who suffering from diabetes mellitus? Of course we do have some portion of people who suffer from diabetes by genetic reasons and by some independent of that subject reasons. But if we'll talk about again the same USA, a lot of people here suffering from diabetes because of incorrect diet. They prefer fast food, they prefer a lot of foods. They don't want to control diet. They are free in their choice and their life, and they just like to eat a lot. And of course, Protestant people say some subject just too lazy to control its diet, or it have some other reasons. But the main problem of that guy is incorrect diet. All that subject need to do is to correct diet. Take less food, take more correct food, more healthy food. But they just don't want. They like to have their way of life. And just because some guy like to eat a lot of burgers, we need to kill animal to give a chance to that subject to have a normal glucose level. Is it correct to kill animal just because some guy want to eat more than he should or she should? And of course, that argument is very reasonable. It is correct. It's not very right thing to kill animal just because of some guy problems. And that problem was very serious. A lot of protesters, a lot of companies, a lot of societies protested against that use of that type of insulin. And nowadays, the problem is easily solved thanks to transduction, thanks to genetic engineering. The main idea, we need to get some insulin. Synthesis of insulin is too expensive. Molecule is difficult. It's difficult to control activity of insulin. It's difficult to synthesize that molecule. It is possible, of course, but extremely expensive. And nowadays, we know property and we know rules of organization of genetic material. We know that every property, every molecule in our body is coded by genes. And people identified gene in our human DNA. which is responsible for structure of the insulin. It is a hormone. It is a simple, by nature, it is simple molecule of biological hormone, and that molecule can be secreted, can be produced or synthesized, of course, not only by human. Multiple other organisms can synthesize that molecule because it is coded by the same DNA which is present in any live organism, including bacteria. So what was done? Gene coding insulin taken from humans was inserted into bacteriophage by genetic engineering, and that bacteriophage can infect E. coli and insert the gene in active part of bacterial chromosome. And when bacteriophage will establish that latency lysogeny, gene of bacteriophage became part of bacterial genome. Bacteria uses that area to synthesize some active molecules, enzymes or some other. And now bacteria have gene of human insulin, but of course bacteria think that it is something belong to them. It is part of their own genome. And E. coli will use that gene, it will produce, synthesize human insulin. So mechanism is very easy, it's very simple. Bacteria just producing insulin instead of some its own enzyme. All you need to do is support the life of that bacteria. And what is required to cultivate Escherichia coli? Agar, water, and sugar. All components are very cheap. All you need to do is to give proper conditions, and bacteria will grow, it will multiply, it will secrete a lot of human insulin. So all you need to do is take insulin and purify. Extract bacteria, remove bacteria, insulin is here. Method is very cheap. By price, it is highly available because bacteria is multiplying independently. You don't need to buy and refresh that bacteria regularly. Agar is cheap and available. Sugar, water, you are welcome. So price of that insulin is very reasonable. It is cheap. In the contrast with previous types of insulins, that one is cheap. Everyone is safe. You don't need to kill any animal. No one cares about bacteria. So nowadays, if you will go to pharmacy and ask for insulin, almost everywhere, 99 and even more % of cases, pharmacy doctor will give you human genetically engineering insulin. It will be written anywhere in the pack and instruction that insulin is obtained with the help of genetic engineering, and by nature that insulin is human. Everyone is happy, no allergic reactions, activity is the same as in our insulin because actually it is our insulin, and price is very available. So transduction mechanism or process of exchange of genetic material in bacteria occurred naturally, but as well used by genetic engineering to produce some drugs, vaccines, and other compounds for our human purposes. Third mechanism, third type of exchange of genetic material in bacteria, it is transformation. Transformation, it is a process by which foreign extracellular genetic material is taken up by a cell. Means external DNA will be acquired by bacteria. And again, some of you can ask, how is it possible to have DNA external? How DNA can exist without a host cell? Of course, it cannot. Any DNA should be released by some other bacteria. And here we have two options. First of all, of course, we all know that bacteria is present in high amount, in high quantity around us. And some bacteria are dying, some new bacteria are appearing. And when bacteria will die, mainly lysis of the cell will occur, and genome of that cell will be released into environment. And of course, some debris of DNA can be acquired, can be grabbed by surrounding cells. And the DNA portion of previous bacterial DNA can be incorporated into bacterial chromosome. and usually it is occurred by breakage and insertion process. As you can see in the right upper corner of the slide, double-stranded bacterial DNA, it will be cut in some one portion, became enlarged, and in that empty area, new portion, new part of DNA will be inserted. Then two ends will be sealed, will be repaired, and bacteria will still have one chromosome. It cannot, of course, have two chromosomes, but that chromosome became bigger, and new properties will be acquired by that bacteria. That type of transformation is stable because that chromosome will remain with bacteria till the end of the day. And the genetic change in transformed cell will be stable. Second type of transformation, second possible type, it is acquisition of plasmids. Plasmid, it is extra chromosomal double-stranded DNA. And plasmids can replicate independently of bacterial chromosome. They use simultaneously because they all present in one bacteria, and bacteria can use both chromosome and plasmid. But replication of chromosomes is independent, and replication of plasmids is independent of chromosomes. And plasmids can be released by alive bacteria into environment, and plasmids can be shared between population of bacteria. As well, if bacteria will be killed and lysis will occur, all container plasmids will be released in the environment, and new bacteria can acquire those plasmids. So that process when foreign extracellular genetic material is obtained by new alive bacteria is known as transformation. If we will compare, for example, chromosome and plasmid, both are presented by double-stranded DNA. Plasmid, of course, is much smaller in size, and usually plasmid contain only few genes with a specific function. And the main amount of DNA as plasmid is presented by non-coding DNA. Means it is nothing, it is empty, it is just some mass of nucleic bases which have no information. But few genes will be useful. And plasmids are divided into two major groups: conjugative plasmids and non-conjugative plasmids. As you know, bacteria can exchange genetic material by conjugation. So conjugative plasmid, it is a plasmid which contain transfer genes that facilitate the beginning of conjugation. If that plasmid can initiate process of conjugation, it is conjugative plasmid. If plasmid can be transferred only with the help of conjugative plasmid, but it cannot initiate process of conjugation, it is non-conjugative plasmid. So it is a property of the plasmid, not a function, but it is a property. According to property of ability to begin conjugation, plasmids are divided into conjugative and non-conjugative. But both types of plasmids will code for additional function. Of course, conjugation is not the only property plasmid is coding. So according to functions, plasmids are classified into several groups. One is fertility plasmid or F plasmid. F plasmids contain F factor, and those plasmids can be inserted into chromosomal DNA. So fertility factor, as you remember, it is a sequence of DNA which is coding for F pylate. And of course, bacteria containing F plasmid will always act as a donor bacteria, or it can be marked as F positive or F plus bacteria. Bacteria which don't have F factor will be marked as F negative, or it will act as a recipient bacteria. So when F positive bacteria will conjugate with F negative, after that conjugation, both bacteria always will become F positive. So that F factor is always shared between donor and recipient. It is multiplying, and after conjugation, both bacteria will become F positive. And that F factor and F plasmids themselves, they are too big in size. They're very big. They're coding for multiple properties. They have more number of genes than usual. And only one F plasmid can exist in each bacterium. It's not possible to have two. Always one fertility plasmid is present per one cell. Next type of plasmid is R plasmid or resistance plasmid. As it is obvious from the name, R plasmids contain genes of resistance to antibiotics. And we talk already, and always I remember you, I remind you, and I always telling you that R plasmids, as any other type of plasmid, can be easily shared between different bacteria. Plasmids are common for any species, any genus, any family, any type of bacteria. It is simple molecule of DNA. Any microorganism can easily acquire plasmid. They don't have species specificity. So when you prescribing antibiotics, of course, your normal flora will always deal with that antibiotic. And your normal flora can become resistant to antibiotic by acquisition of R plasmids, by other mechanisms, by bacteria will have and it will keep information about how to resist to that antibiotic. And years, decades after the case, you can forget that you took that antibiotic, which antibiotic you took, but your normal flora will remember, partially because of R plasmids. And when another pathogenic bacteria, now pathogen, will come to your body and cause some infection, initially when doctor will take samples, or maybe you will take sample from subject, you will perform identification, you will identify species of bacteria, you will perform antimicrobial susceptibility testing, you will identify which drugs are active, which are effective. You will prescribe the drug, but in a few days you will see that your patient don't become better. And when you will repeat the sample and repeat the test, you will identify that actually your bacteria is now resistant. And if you will ask how it is possible that microorganism in a few days became absolutely non-sensitive for your drug, possible answer is R plasmids, which were present in your normal flora bacteria, and now they are shared with pathogenic microorganisms. R plasmids, as in any other plasmid, they can be excreted and surrounding, and it can be secreted by one bacteria, your normal flora bacteria, but it can be acquired by another bacteria, pathogenic bacteria. And previous use of antibiotics, especially recent use, it is always the high and important risk factor of resistant infection in future, in closest future. So again, please use antibiotics only when they are really required and use them correctly. It should be rational use because R plasmids can accumulate in your body and future infections will be much more difficult to treat if pathogenic bacteria will acquire R plasmids which are stored in your normal flora bacteria. So another type of a plasmid, it is a virulence plasmid. Virulence plasmid, it is a plasmid which coding for some pathogenic properties, and previously harmless bacteria will be turned into a pathogen. Mainly those virulence plasmids are coding for some toxin, and they named as tox plasmids. And if plasmid have a gene of some toxin which can harm our body, that plasmid, it is a virulence plasmid, it can be called a tox plasmid. One more group of plasmids, it is a degradative plasmid. Degradative plasmid contain genes for enzymes which are able to digest some compounds. Means that bacteria, for example, was glucose negative previously, but after acquisition of subplasmid, it can become glucose positive. That property of synthesis of some new enzyme coded by plasmid is known as degradative plasmid. Degradative plasmids are conjugative. they associated with F factor. And again, we said about resistance plasmid, I forgot to say that R plasmid is not always conjugative. They can be, but they're not always so. And it is possible to have multiple R plasmids per one cell. And one bacteria can have multiple genes of resistance to multiple antibiotics at the same time if R plasmid is non-conjugative. So degradative plasmids are always conjugative plasmids. And one more type of plasmids, it is a col plasmid, refers to bacteriocin, first bacteriocin, colicin. So col plasmids contain genes to produce bacteriocins. Again, bacteriocin, it is a protein which can inhibit or suppress the growth of closely related bacteria or similar bacteria. And col plasmids are an important factor of resistance to surrounding microorganisms. So initially they are named because of E. coli bacteriocins, but actually they can code for any type of bacteriocin. So those three mechanisms are main mechanisms in bacteria, but they are not all bacteria have, and especially you can ask, what about viruses? Viruses have no active life. They cannot undergo conjugation. Of course, they cannot acquire some foreign material. They're inactive, they're in it. So one more mechanism of evolution in general and in bacteria and microorganisms, especially important mechanism, it is a mutation. Of course, you all know that mutation is a permanent alteration in the sequence of nitrogenous bases of a DNA molecule, which can lead to variations in genes. And that permanent alteration is not always possible, or it should not be possible in human, because we have very strict and we have very good systems and mechanisms of mutation control. Any mutation should be very fast repaired, and sequence should be stable. But in bacteria, that mechanism is absent, and bacteria undergoes mutation always, constantly, regularly, and very frequently. So mutation is a permanent alteration in the sequence of nitrogenous bases of a DNA. Permanent means it will remain forever. Once occurred, bacteria will have that changed or altered sequence for the rest of the days. So uh, term mutation, why mutation? What is mutation? Initially, it was coined in 1886 by Hugo de Vries. It is the term derived from a Latin word mutatio. Mutatio means changing, and mutation, it is a changing. So the process of mutation, that stable process when mutations occur, is known or called as mutagenesis. And we know that mutation can be induced by some agent. And that agent which induces mutation is called as a mutagen. So in genetics, we always have two types of organisms, wild type and mutant type. So initially, in perfect way, wild type, it is natural type of organism which exists around us. And if something occurred, if we induce some mutation experiment, the changed organism will be caused as a mutant. But of course, in nowadays, it's impossible to identify which type of DNA was natural, because nowadays every organism already mutated, and we all are mutants if we will compare our DNA with very initial one. So it's more correct to say that reference strain is called as a wild type. If you took some organism, you studied genome of that organism, that organism will be called as a wild. When mutation will occur, you will compare that mutant with the initial one. That initial should be called as a wild type. So in experimental practice, we have two types of organisms. Wild type organism means before mutation, before current mutation, and mutant, strain, which is after current mutation, during one experiment, during one investigation or observation. So, of course, you all know, but we still will remember and we will remind types of mutations according to mechanism. If you talk about uh, bacteria or even about virus, of course, you all should understand that it's impossible to have chromosomal mutation. If in eukaryotes we say about chromosomal and some point mutations, in bacteria and viruses, all mutations are always limited to one single DNA. It's not possible to exchange different parts of chromosome because in bacteria chromosome is single and it is circular. In viruses, chromosome is absent at all. So all mutations are limited to one molecule of DNA. And of course, again, you all know that mutations can be spontaneous, occurring naturally by errors during DNA replication, or they can be induced by a mutagen in an environment or in experiment with different multiple purposes. But all mutations have known and fixed mechanism. So first and the most frequent mechanism, it is substitution. Substitution with one nucleotide with another nucleotide. And substitution is always point mutation. Again, it is not possible to have substitution of one part of the chromosome with another. It is always point mutation. It is limited to one only nucleotide. So substitution in microorganisms involves changing of a single base in the DNA sequence. One will be changed. We have two types of substitution mutations. If one purine or one pyrimidine nucleotide will be replaced with other, means AOG or CNT, that mutation is called transition. If a purine nucleotide is replaced by pyrimidine or vice versa, that mutation is called transversion. Again, substitution, both transition and transversion, is the most common mechanism of mutation. So, second mechanism of mutation is known as deletion. Deletion, it is when one nucleotide is removed or thrown out of DNA chain. In that case, number of nucleotides will decrease. Another similar mechanism, it is addition, when one new nucleotide is accidentally added to a chain of DNA, and in that case, DNA will become longer. So again, type of additional mutation, kind of it is transposon. When transposons are inserted into genes, again, it will lead to disruption of the gene. It is insertional mutation. It is kind of additional genes, but it is not a single nucleotide. So it is another type of mutation known as insertional mutation, when transposon jump in gene is added to gene. So if we will talk about result, of course, we can understand mechanism, but we need to know how is it important, what will it lead to. So according to a result of mutation, we have again classification, second classification of mutations. Initially, you all know that our DNA and bacterial DNA as well is presented as a number of triplets. Every triplet is coding for one amino acid. Some sequence of amino acids is coding for one protein. One protein is responsible for a property, and every protein is limited by start and stop codon. And we have property which is coded by genome. So, of course, if mutation will occur in genome, it should have some reflection in final product. And according to that change in final product, all mutations are classified into several types. So, for example, I took some accidental sequence of DNA, which is coding for known amino acids. It is our wild type. It is organism before any mutation. So now let's pretend that mutation occurred. So we will talk about point mutations and, for example, some replacement of one nucleotide occurred, and we have missense mutation. Missense mutation, it is mutation which leads to change in amino acid sequence when one wrong codon and one wrong amino acid appeared in final product. So one amino acid is changed. The total length of the protein product remains the same. It still should have start codon, stop codon, it will be the same number of amino acids, but one is changed. As far as structure of final product is very similar to initial one, that product can have some function, even very similar function. It can act, it can have some efficacy, but of course function will be altered. And missense mutation again manifests by changing amino acid sequence, but final product will be produced, it will be synthesized, it will be very similar to initial one. Next type of mutation is nonsense mutation. Nonsense mutation, it is mutation which leads to formation of a stop codon instead of some amino acid, and that mutation will causes termination of protein synthesis. Of course, final product will be incomplete. And according to place of mutation of that mutation, final product can be almost identical if its acute mutation occurred in some end of the sequence. Protein can be very similar to initial one, and again, it can have some property, it can have some activity. But if that mutation occurred in the center or closer to start of the final product, of course, the product will be abortive, it will be very short, and function will be lost. And we have silent mutation. Silent mutation, it is a single substitution in DNA-based sequence, which will result in appearance of a new codon, but which is still coding for the same amino acid. And of course, you all remember that that some amino acids can be coded by several sequences. Several combinations can code for the same amino acid. So if mutation occurred, but that codon is still coding for the same amino acid, final product will not be changed. It remains absolutely the same. And mutation occurred, but result will not be visible. Nothing will be changed for that organism. That's why mutation is called silent mutation. So one more, actually, again, usually it is a result of addition or deletion, but we have frame shift mutation. That mutation is more important, it is more difficult because I hope again you remember what is frame shift. It is ability of organism to recognize triplets. Of course, we have some DNA, it is accidental, it's sequence, some sequence of nucleotides, but we should recognize correct triplet. And if you will use correct identification, our DNA can be used. If some change will occur, if we will not be able to recognize, for example, in that picture, AUG is the start codon. If we will read, if our enzymes will read AUG as AUG, it will be the start of the gene. But if it will be some, for example, UGC, if that frameshift will be wrong, then of course we will have absolutely incorrect information. We will not be possible, we will not be able to use our DNA. So that ability to recognize correct triplets is known as a frameshift. So if addition or deletion, for example, occurred, of course, one nucleotide is removed. And in that case, the whole reading frame will be damaged. Shift in the reading frame will occur. And for example, here in that slide, start codon is recognized correctly, gene will be used correctly, but one nuclear basis is absent, and further combinations will be recognized incorrectly. Instead of CAU, first codon will be recognized as AUC. And of course, it is absolutely another amino acid. And the whole sequence, the whole further sequence will be changed. All amino acids after frameshift mutation will be recognized incorrectly. And even stop codon will be placed in incorrect position. Of course, overall sequence of a gene and overall sequence of amino acids will be altered, and final product will be absolutely wrong. It will have wrong amino acids, wrong sequence, wrong length, and it cannot be used. Properties, multiple properties even can be lost. And reading frame should divide nucleic acid sequence into consecutive non-overlapping triplets, coding for one action. If the sequence is altered, then of course triplets of nucleotides will code for wrong action, wrong product, and the whole further properties will be lost by bacteria. So according to result, again, mutations can have some benefits, or maybe it will have some penalties. Again, according to in which conditions which property is changed. Some mutations can be useful. They can bring new important property which is essential for bacteria. But if some essential property will be lost due to mutation, of course, that mutation can lead to loss of the property or even death. And if mutation is leading to death, it is known as lethal mutation. After mutation, the bacteria will not be viable, it will die. And we have conditional lethal mutations. Conditional lethal means that mutation will become lethal only under certain conditions. For example, if bacteria initially, wild type of bacteria had enzymes to utilize glucose and lactose, bacteria now is present in lactose medium, lactose-containing medium, mutation occurred in glucose gene, and bacteria lost the property to utilize glucose. But while bacteria remain in lactose environment, it will remain alive. When bacteria or environment will change, bacteria shifted to glucose-containing medium, bacteria will die because now it don't have enzymes to utilize glucose. And mutation will not lead to a death immediately, only under some circumstances. And that type of mutation is known as conditional lethal mutation. So if you talk about genetic material of an organism, it is known as a genotype. Genotype, it is a complete set of genetic determinants of an organism. And we said before that in medical practice, it is enough to know species of bacterium. For doctors, it is sufficient. But of course, we said that species can be classified or subdivided into much smaller groups according to different properties. And one of that example, it is a genotype. Of course, genotype is much narrower group than a species. and every species of organism can be classified into genotypes because genetic or set of genetics determinants of different organisms within one species can be absolutely different. Again, it is important in genetic engineering, it is important in experimental microbiology, but not so important in medical practice. But we have the genotype. Of course, you all know that genes are coding for properties which should be manifested in real life in obvious characteristics. So phenotype, it is direct reflection of a genotype. And phenotype, it is an observable characteristics of an organism that results from interaction of its genotype with environment. Genome is interacting with environmental conditions, and properties, observable properties, are known as a phenotype. So every property of organism initially is coded by a gene. If mutation will occur, again we said that it can lead to changes in phenotypic properties as well. But in the case of bacteria as well as the case of any live organism, we can have another unique type of changing in properties, which is known as modification. Modification, it is phenotypic changes of one or few characteristics not accompanied by changes in primary DNA structure. Means genome remains the same, but observable characteristics are changed. Modification can be of two main types, morphological and biochemical. Morphological means that morphology of bacteria will be changed. Some structural organization is changed. And one of the example, it is loss of motility by bacteria when it is placed in the formalin. Genome will remain the same, genetic composition is absolutely the same, nothing occurred. But phenotypical properties are changed. Or in the case of biochemical modifications, again, the perfect example is inducible synthesis of enzymes. Very often bacteria trying to save energy, and it will not synthesize enzymes if sugar or if target for that enzyme is absent. Again, if bacteria is present in lactose media, for example, it will produce enzymes required for utilization of lactose, but the rest enzymes will be switched off. They're not required now. But if environment will be changed, you place bacteria in glucose medium. Bacteria can induce synthesis of new enzymes. So if you will check initial bacteria, it will be glucose positive, glucose negative, sorry, but lactose positive. But when you will transfer it to glucose containing medium in some time after that, bacteria will become glucose positive. So it is a type of, again, modification, biochemical, but modification, because genome remains the same, but properties, observable characteristics are changed, phenotype is changed. And one example of that changing of properties without changing its genome is adaptation of bacteria to different hosts. And that classification is known as a biotype. Again, biotype of bacteria is phenomenon when identical microbes have different phenotypical characteristics. And one of the examples, for example, Brucella. It is, nowadays, it is suggested to be the only species Brucella meritensis, but it has multiple biotypes. Actually, initial host of that bacteria is the key for that classification. And if Brucella is obtained from dogs, it is Brucella canis. If it is obtained from sheep, Brucella ovis, suis, microti, and a lot of biotypes we have. Actually, by all properties, bacteria are very similar, but they have different biological hosts, and it is the key to classify one bacteria to different multiple biotypes. So again, in medical practice, biotypes can be important because it is important for identification of initial host. When disease is zoonotic, originate from animals, we need to know source of infection. And identification of biotype, of course, is the key to identify that source. So in your exams, you have examination questions. You have two questions. In examination tickets, you have two questions: inheritable variability and unheritable variability. And it always confused you asking, what is it? I don't know. What is inheritable? What it means? So if changed, if some new property, that new structure of microorganism able to be transmitted from maternal cell to daughter cell, it is inheritable. It means it can be transmitted from one generation to other. And as we said, it should be alteration in genome. It is a mutation. If mutation occurred in one cell, it will be delivered to daughter cells as well. It is inheritable variability. Unheritable means the change is not able, is unable to be transmitted from maternal cell to daughter cell. And modification is the type of unheritable variability, because genome is the same, but manifestation, phenotypic manifestation, depends on environmental condition. And new cell will receive only genome, and phenotypic manifestation will depend on environment. So that change cannot be transmitted to new generation, and that one is unheritable. So don't be confused, it's very simple: in mutation and modification. That's actually all we need to hear about, from you on our exam.